Hello friends, I am Harun and it's my YouTube channel Harun Biozone. Today in this video I will discuss about a total concept of nitrogen fixation process like ammonification, nitrification, nitrogen assimilation, denitrification, all. Okay, so in this video I will discuss a complete course of nitrogen cycle or nitrogen fixation process. So don't be late, let's go. First, nitrogen. Nitrogen, the movement of nitrogen between atmosphere and the living organism in different forms is called nitrogen cycle. What is nitrogen cycle? When nitrogen is moved from atmosphere to organism body, this is called nitrogen, and again organism to atmosphere, this is called nitrogen cycle. Why nitrogen cycle is very important? Because in the atmosphere there are 78% nitrogen present, okay, but no plant and animals, some plant except some plant, but mostly plant and animal do not accept it from atmosphere. Okay, atmosphere 78% নাইট্রোজেন থাকলেও সেই নাইট্রোজেনটা উদ্ভিদ বা প্রাণীরা অ্যাবজর্ব করতে পারে না, যদিও নাইট্রোজেন ইজ দ্য স্ট্রাকচারাল অ্যান্ড ফাংশনাল ইউনিট অফ human body or of organisms body. So nitrogen is essential for this process. The nitrogen is converted into ammonia, then nitrate and nitrite. Okay, then again denitrification process, nitrogen return to the atmosphere. The plant absorb the nitrate in ammonia form, nitrite form they absorb, and from plant we collect this nitrogen. There are different processes of nitrogen fixation present in atmosphere. First of all, the convert the nitrogen. The overall process of nitrogen fixation is to absorb one molecule of nitrogen require 8 proton, 16 ATP and 8 electron to produce 2 molecule of ammonia and generate 16 ADP and 16 phosphate. So to absorb 1 molecule of nitrogen require 16 molecule of ATP. Clear? The animals that absorb nitrogen is called diazotroph. Okay, the diazotroph are organisms that fix atmospheric nitrogen, and in the presence of the enzyme nitrogenase. Clear? There are some free living bacteria like Klebsiella, Azotobacter, cyanobacteria, and some symbiotic bacteria like Rhizobium absorb atmospheric nitrogen. They fix near about 1011 kg per nitrogen per year. Okay, about 60% of Earth's fixed nitrogen absorbed by this organism. Lightning and ultraviolet radiation. Lightning during rainy season, lightning and ultraviolet radiation also fix 15% nitrogen in the atmosphere, from the atmosphere to the earth. Ammonia can also be obtained by reduction of nitrate that is present in the soil. So ammonia convert into nitrate, and this nitrate that absorbed by organism. Nitrate reduction can be carried out by most plant and organism. The ammonium resulting from these two processes can then be assimilated by all organisms. Okay, so here some typical process. During lightning, the nitrogen converted into nitrite and nitrate salt. During when the other organism are dies, plant and animal both, some decomposer converted to ammonia, they fix into the plant. Okay, some ammonification and assimilation. Let's go. The classification of nitrogen fixation. There are three processes of nitrogen. First, atmospheric nitrogen fixation, industrial nitrogen fixation, and biological nitrogen fixation. The industrial nitrogen fixation is man-made process. Man-made artificial laboratory process. It is required for the production of ammonia to produce different type of fertilizers. Atmospheric nitrogen directly fixed by plants during rainy season. Or biological nitrogen fixation, there are different type of biological organisms that sometimes forms nodules, sometimes non-nodules, sometimes symbiotic, sometimes free living organism fix atmospheric nitrogen. First one, atmospheric nitrogen. Atmospheric nitrogen fixation mainly occur during rainy season, because in this time lightning occur, and in the presence of lightning the atmospheric nitrogen combine with oxygen to produce nitric oxide. This nitric oxide again combined with oxygen by oxidation reaction produce nitrogen peroxide. This nitrogen peroxide and H2O combine to produce nitric acid and nitrous acid and nitric acid. This nitrogen dioxide and water and oxygen produce nitric acid. Okay, calcium oxide plus nitric acid to calcium nitrate, and this is salt. They absorb plant in this form. Nitrate, nitric acid and ammonia produce ammonium nitrate, and nitrous acid plus ammonia produce ammonium nitrate. In this form the plant can absorb this atmospheric nitrogen directly. In this case, no microorganism take part for the absorption or fixation of atmosphere. Next question, next topic is industrial nitrogen fixation. This is man-made process. In this process, we especially use the Haber-Bosch process. Here, the nitrogen taken directly from atmosphere, and the hydrogen produced by methane reforming process. Okay, in operating condition, the nitrogen and hydrogen in the presence of high temperature 400 to 500 degrees centigrade temperature, and in the presence of 200 to 400 atmosphere, 150 to 400 atmosphere pressure, the nitrogen and ammonia in the presence of catalyst like potassium hydroxide and aluminum oxide produce ammonia, and this ammonia is collect and use in fertilizer. Like then biological nitrogen fixation. এখানে দেখতে পাচ্ছ biological means the nitrogen fixation occur in the presence of some microorganism. Biological nitrogen fixation is a main process of nitrogen fixation in earth. There are three types of nitrogen fixation: biological nitrogen fixation, non-symbiotic or asymbiotic biological nitrogen fixation, associative biological nitrogen fixation, and symbiotic nitrogen fixation. First, non-symbiotic. Non-symbiotic means here the microbes are free living. Soil contain number of free living nitrogen fixing organisms, and they are free living in soil, do not associate with roots of plants. Some free living anaerobic bacteria, these examples are very important. Okay, there are some free living aerobic nitrogen fixing bacteria like photosynthetic Chlorobium chromatium, non-photosynthetic Azotobacter, Azomonas, Daxia, Beijerinca. Free living anaerobic nitrogen in the absence of oxygen, the fixed atmospheric nitrogen. Photosynthetic Rhodospirillum, non-photosynthetic Clostridium. Free living chemosynthetic bacteria Okay, heterotrophic disulfivibrio, cyanobacteria, blue-green algae like heterocyst bearing nostoc, Anabaena rivularium, Carothropus, and non-heterocyst forming Oscillatoria, Gliocapsa, Lingbia, Plectonema, free-living fungi like yeast and Pullularia also fix atmospheric nitrogen. The asymbiotic free-living nitrogen fixing are quite primitive. This is the not pioneer process. It is less effective, but they fix atmospheric nitrogen when present in free-living condition in soil. They convert same process nitrogen to ammonia, ammonia to nitrate, then nitrite. Okay, they fix in this form. Associative symbiotic nitrogen fixation. Associative means they do not symbiotically present, but the bacteria present in the rhizosphere region of root. Rooted চারিদিকে তারা থাকে. They are called associative microorganism. Okay, certain bacteria live in close contact with the roots of cereal and grasses. তারা কি করে, সেই রুটের রাইজোস্ফিয়ার রিজনে খুব পাশাপাশি অবস্থান করে এবং তারা কি করে, নাইট্রোজেনকে ফিক্সিং করে. ইট ইজ কল্ড অ্যাসোসিয়েটিভ সিম্বায়োসিস. The bacteria reside in the transition zone between soil and root. Suppose this is root, and this is soil, and they present between each transition region present around the root here. Sometimes enter the root. Some they fix at nitrogen is absorbed by the roots and turn the bacteria get nourishment from the carbohydrates by the root. So these bacteria fix nitrogen to root and provide carbohydrate nourishment. Example, Azospirillum brasiliensis, it association with cereal roots. Okay, Beijeringia association with the root circle, and Paspal association with the tropical Paspalum nota. Then symbiotic nitrogen fixation. Symbiotic means here microorganism present inside the root, here roots of the plant. Symbiotic nitrogen fixation is a part of mutualistic relationship which plants provide niche and fix carbon to bacteria and excrement. They are present inside the roots and they fix atmospheric nitrogen. The nitrogen fixing through nodule formation by formation of nodule. Okay, this is a root here, a root secretes some flavonoids, then nodulation gene activated and node factor, bacteria is node factor. This node factor help the bacteria enter into the root here and formation of nodule. Okay, example, Bradyrhizobia japonicum and Azorhizobium colinadenus. The step-by-step process is first the chemical signal. The root secrete, first of all, the root secrete First, chemical signaling. Chemical signaling is the process. Chemical signaling means root secretes some chemicals or flavonoids. The legume root secrete compounds called flavonoids, okay, into the soil which attract the bacteria. First, root secrete some flavonoids. Flavonoid attract the rhizobium bacteria. Then root curling occur. When the rhizobium bacteria are attached to the rhizosphere region, the root curl and bacteria enter into the root curling starts. Infectious thread and primordia. The bacteria penetrate the root here. After penetrate the bacteria into the root here, they produce infectious thread are produced. Okay, a tube that delivers the bacteria deep into the root cortical cell. Infectious thread is one type of hyphae-like structure that enters the bacteria deep of the cortical cell or cortex region. Okay. Bacterial differentiation. The bacteria enter the plant cell and transform solely in branched non-dividing structure called bacteroid. এবং inside the root cortex they produce solen ফোলা ফোলা ধরনের ব্রাঞ্চ শাখা প্রশাখাযুক্ত নন ডিভাইডিং স্ট্রাকচার এবং এটাকে আমরা বলছি কি ব্যাকটেরয়েড। এটাকে বলা হচ্ছে ব্যাকটেরয়েড বলা হয়। This is bacteroid. Okay, this is bacteroid, and then the bacteroid produce the enzyme nitrogenase. The bacteroid, first plant secretes some flavonoids, attract the bacteria. Bacteria come into the rhizosphere region, then bacteria penetrate into the cortex region and form an infectious thread that is called bacteroid. Bacteroid produce nitrogenase enzyme that convert atmospheric nitrogen to ammonia. But there is a problem. Nitrogen is a oxygen sensitive enzyme. In the presence of oxygen, they inactive. So do not fix atmospheric nitrogen. For this purpose, the plant produce a leg hemoglobin. Leg means leguminous plant. Hemoglobin is blood-like substance or pigmentation. Pink iron containing pigment called leg hemoglobin. This leg hemoglobin protect the nitrogenase from oxygen. Okay, so leg hemoglobin called oxygen scavenger. Then nitrogen fixation through nodule formation in non-leguminous plants. Okay, some bacteria produce nodule formation also occur, but this process occur in non-leguminous plant. Okay, non-leguminous plant like genus Frankia. Genus Frankia forms root nodule in Alnus species. This is the Alnus species, and the Frankia produce produce nodule. Okay, rhizobium also root nodule in genus Parasponia. Leaf nodules are formed bacteria Klebsiella in genus Psychotria and by bacteria Burkholderia in genus Pavetta Zimmermannian. Okay, these are non-leguminous plants where nodules are formed. Nitrogen fixation through non-nodulation, but here nodule do not form in this process. Say for example, lichens. Lichen is a symbiotic association of algae and fungus. They fix atmospheric nitrogen when the algae are cyanobacteria or green algae. Anthocerus, a bryophyte associated with nostoc and fix atmospheric nitrogen. Azolla a fern associated with anabina. This is the azolla leaf, and anabina present inside the azolla leaf. Okay, for Anabini azoli. Okay, they fix atmospheric nitrogen. Cycas, a gymnosperm in association with anabina or nostoc, blue-green algae, it is called coralloid root and they fix atmospheric nitrogen. Gunnera, microphylla, angiosperm associated with nostoc in its stem. Okay, roots of Digitaria, sorghum, and maize associated with spirillum. Symbiotic diazotroph means the organism that fix atmospheric nitrogen are more advanced and efficient. This type of nitrogen fixation is more advanced and more efficient 100-200 times than the asymbiotic diazotrophs. Then ammonification. Ammonification means when the plants and animals are die, or here they decompose by bacteria and fungus to produce ammonia. Okay, this ammonia fix atmospheric nitrogen ammonia. Bacteria carrying out ammonification are called ammonifying bacteria, like example Bacillus, Proteus, Clostridium, Pseudomonas, Streptococcus, Micrococcus. Or these bacteria also fix atmospheric nitrogen into ammonia and convert it into ammonia. Like first nitrogen, this is the nitrogen, they absorb hydrogen and form hydrogen then again absorb hydrogen and form NH3 and NH3 or 2NH3. Then again hydrogen absorbed to produce 2NH4 plus ammonia. In this process, this bacteria also absorb atmospheric nitrogen. Then one process is decomposing process, by which the plants animals when die, they decompose by bacteria and fungus and convert into ammonia. Another process is the nitrogen in the presence of hydrogen ion convert into ammonia. Okay. Next, nitrification. Nitrification, there are two steps. Ammonia convert into nitrate and nitrite. Here ammonium. There are two processes. First, ammonia in the presence of oxygen and nitrogenase enzyme also. Ammonium monoxygenase, single nitrogen, and in the presence of nitrosomonas bacteria, they produce nitrogen dioxide plus 3 molecules of hydrogen, or 2 electron produced. But step 2, nitrogen dioxide, again nitrate oxide, in the presence of nitrate oxidoreductase, again water in the presence of water molecule convert into nitrite, that is nitrite, and 2 molecule of hydrogen electron. So in ammonia to nitrate convert by nitrosomonas bacteria, and nitrite to nitrate by nitrobacter. This is the major step process, important steps for nitrogen fixation. Now, most important point, nitrogen fix. Okay. Then what is the next function of nitrogen in organism body? This is nitrogen assimilation, because nitrogen convert into ammonia. And ammonia is a highly toxic material, and this ammonia also produced by plants in photorespiration. Okay. So to remove this high toxicity, this ammonia enter into the this ammonia mixed with amino acid. Okay. To avoid this toxicity, ammonia combined with amino acid to produce protein. Here, this is the process in plant during photorespiration. The nitrate convert into ammonia in the presence of nitrate reductase and ferredoxin. The nitrogen assimilation process is occurred by GS-GOGAT system. Look at this picture. This is the plastid where photorespiration occurs, and this is the cytoplasm of the cell. In the cytoplasm, the nitrate in the presence of nitrite reductase convert into nitrogen dioxide, again nitrate reductase to produce ammonia, or plant can absorb ammonia directly from the soil environment. Okay, this ammonia in the presence of glutamine synthase on with the help of ATP produce glutamine. This is on amino acid. Here the glutamate amino group, the overall reaction is here. This is the ammonia, and combined with the amino acid glutamate in the presence of ATP and glutamine synthetase enzyme, they produce glutamine and 2-oxoglutarate. 2-oxoglutarate means alpha ketoglutarate. Clear? Again, in the plastid where ammonia is produced by photorespiration, then against it the presence of glutamine synthetase enzyme convert glutamine, and glutamine again in the presence of glutamate oxoglutarate amino transferase. Glutamate oxoglutarate aminotransferase enzyme, this glutamine convert to oxoglutarate into glutamate. This glutamate again convert into glutamine. Okay. Here 2-oxoglutarate in the GOGAT, glutamine oxoglutarate aminotransferase means that amino group is in the presence of here two types of the GOGAT system occur. One is NADPH dependent, another is ferredoxin dependent. Two type of reaction occur and produce the glutamate. Clear? This is the main pathway of nitrogen assimilation. The another pathway is glucose glutamate dehydrogenase pathway. Here the 2-oxoglutarate or alpha ketoglutarate in the presence of ammonia. They attach with the oxoglutarate and produce the glutamate. Produce glutamate. Okay. Here ATP is not required, but this is a NADPH dependent process and depend on the glutamate dehydrogenase. So where the ATP is unavailable, only ammonia available, this process is efficient. This is the alternate pathway of nitrogen assimilation. Another pathway also present, that is called asparagine synthetase. Asparagine synthetase pathway, where the glutamate that carry on amino group carry, and in the presence of oxoglutarate, the glutamate convert into aspartate. And 2-oxoglutarate also produce in the presence of aspartate aminotransferase. So this amino group transfer from glutamate to aspartate. Convert aspartate to the glutamate to aspartate. Okay. Again, in another reaction where the glutamine and oxoglutarate, where the glutamine is in the presence of aspartate, glutamine and aspartate, the amino group of glutamine is transfer to asparagine, to glutamine to asparagine, and glutamate also produce in the presence of enzyme asparagine synthetase. So in case of aspartate aminotransferred, glutamate converted to aspartate, amino group transfer from glutamate to amino group. These all products occur in the Krebs cycle of photorespiration, Krebs cycle of respiration. So in this way, glutamate to glutamine, glutamate to aspartate, by aspartate to asparagine process, the amino group or amino group that absorbed by nitrogen fixation is converted onto another amino acid and produce different type of protein. Okay. Last process is denitrification. After when the organism dies, the enzyme, the atmospheric fixed nitrate or NO3 form is converted into nitrogen gas and reversed to the atmosphere. In this process, first NO3 minus, nitrate salt, converted into NO2 minus, then NO2 minus to NO, and NO again in the presence of 2 proton and 2 electron, N2O plus S2O. N2O 2 proton plus 2 electron produce nitrogen gas and back to the atmosphere. The overall reaction is 2NO3. Overall reaction is 2NO3 plus 10 electron plus 12H plus convert into nitrogen gas and 6H2O. This process or denitrification process occur by some bacteria and in the presence of enzyme reductase. Okay, it is a reduction process where the micrococcus denitrificans or thiobacillus denitrificans or some pseudomonas, acrobacter and serratia species also take part for denitrification reaction, or by denitrification the atmospheric nitrogen again return to atmosphere and balance the nitrogen gases in atmosphere and earth. What is the significance of nitrogen? First, nitrogen is the major structural and functional unit of organism's body or human body. Okay, so but atmospheric human do not accept atmospheric nitrogen or fixed atmospheric nitrogen. So nitrogen fixation process is very crucial for every organism, plant and animals, because by nitrogen produce amino acid, produce protein, nucleic acid, DNA, RNA. It is the building blocks of life. All organisms require nitrogen to live and grow. Nitrogen is a key part of amino acid and nucleic acid, and also an important part of ATP, which is the basic energy molecules for living things. I think you can understand the overall nitrogen fixation process and different steps clearly. So I think this video solved, because from nitrogen fixation at least one MCQ question are coming in every competitive examination. So I think this question you can solve easily after watching this video. Thank you for watching this video. Thank you.